I'm Lily Fountain, Chair of the Maryland Natural Places Committee. Welcome to our talk tonight with Jim Mccain from DNR on the status and distribution of Maryland mussels, which is really exciting to learn what a sentinel species they are for our state and what we can do to help keep them around. I'm going to go ahead and introduce the chair of the Endangered Species Committee, which is Mark Inlay, and he's going to tell you about his background with mussels, which is quite extensive, and then introduce Jim.
Okay, thank you, everyone. Jim asked me to talk first when he heard about how 5 decades ago, for a whole decade, all I did was work with freshwater mussels. It started, I got my PhD, I worked on how they know what time of, whether they, how do they decide what time of day it is to do their thing. It was fascinating. And then I got a federal job at the Duluth, Minnesota, the Federal Water Pollution Control Administration. It was the first federal agency to determine what toxic levels of hydrogen, nitrogen, phosphorus, zinc, copper. And I decided I love the mussels around there, and I collected some from a local stream, and I brought them in and used them for testing. And then my family, Alice and the kids, said, "What the heck? You haven't been coming home every night, 3 hours late every night for 3 months." I had an idea. Instead of just testing how much kills them, I decided to test how much prevents them from growing and reproducing. And my method worked. I tried a wild idea out and it worked. And after 3 months, my boss says, "Hey, keep doing this." And now, and I started the movement going, don't just see what prevents species from dying, but also what prevents them from growing and reproducing. And then I found so many species of freshwater mussels were endangered, so many. And I was in the Wisconsin, northern Michigan area, but all over. I decided I got to do something. And I transferred as a federal employee to the new agency called Office of Endangered Species in Washington, D.C. And in 10 years, I listed about 24 species of freshwater mussels as endangered. And one of them, Jim is going to love talking about tonight, Elasmodon heterodon, the dwarf wedge mussel. And by the way, you citizen scientists, if you go by a river, it's right there at the edge where the water's only a foot deep, so you don't have to get all wet. And I used it to stop a terrible federal pollution problem at McIntosh Run, a development project. So anyhow, may I introduce Jim Mccain? And Jim, thank you so much for continuing this. I was it good for the plant people to steal me from mussels? I don't know what they did. But anyhow, I'm so glad to be back with mussels. Thank you, Jim.
I just want to welcome everyone who came in during Mark's talk, Jim is pulling up his PowerPoint, and we're happy to have you here. And if you want to put your location in the chat, we'd love to have that. It helps the speaker sometimes to know what parts of the state the people are from. So far, we have at least, I don't know, 10 different counties in Maryland, so it's pretty exciting. I guess I'll mention something. You're going to learn from Jim tonight how amazing mussels are for indicating the health of streams. And we do have a new campaign, Save Our Streams, that just launched tonight. And there's another talk about stream restoration and what are the best ways to preserve our streams. And so you can check out this campaign if you want to sign the letter to prevent the permitting to expand some of these stream restorations. You can learn more about it on Sunday, and you can sign this campaign if you're interested. You ready, Jim?
Yeah, I think so. How does it look on your end, Lily?
Great.
Okay. So first, a quick introduction. My name is Jim Mccain, and I've been working with Maryland DNR Natural Heritage Program for about 25 years. I've been with DNR over 30 years, and I've had a great job, still have a great job, but one of the groups of organisms I've had a chance to work on quite a bit is freshwater mussels. And that's will be the focus of my talk tonight. You can see here that there's a few other folks that I listed as presenters, Matt Ashton, Dan Feller, and Dave Brinker. And really, these guys we've all collectively worked together over the years, working on various aspects of freshwater mussel work in Maryland. And we're particularly lucky to have Matt Ashton on our team. Matt actually went to graduate school and is a malacologist. And, I can say that, through Matt's efforts, we've been able to really ratchet up the work we've been doing with mussels in Maryland over the last decade or so to a much higher level.
Cool.
So what I want to do is just quickly give you an overview of who I work for to try to add some context to this talk. I work for the Maryland Natural Heritage Program, and I'll give an overview of freshwater mussel life history, their status at different levels, talk about their diversity at different spatial scales, and then dive into the mussels we have here in Maryland, what their status and distribution is, talk about some of the threats, and then follow up or close with a few projects that, a few recent projects we've been involved in. So for those of you who are not aware, the Maryland Natural Heritage Program has been around since 1979. We have about 20 staff. And this is the lead agency in Maryland, state agency, that deals with the protection and recovery of rare, threatened, endangered plant and animal species, non-game species conservation, the protection of natural areas. We track the status of over 1,200 rare, threatened, endangered species and some natural community types. We're involved in a lot of inventory, monitoring, and research. And I guess most importantly, we try to involve ourselves as much as possible in trying to protect, restore, and conserve these species and habitats and ecosystems that they're dependent on. So this is just, kind of a who's who. Some of you may recognize some of these people. These are some of our staff and some of the people we've contracted with over the years. The person down the lower right is Chris Fry. He's my, I guess, plant equivalent in our program. Chris has oversight of coordinating changes to the state's rare, threatened, endangered plant list. And I have a similar role dealing with animals. So there's been some really important environmental legislation, over the decades. And of course, some of those deal directly with endangered species. In 1972, a year before the federal ESA Act was passed, Maryland passed its own Endangered Species Act. And it was modeled closely after the federal ESA, which was in draft form at that point. Another important piece of legislation was the Chesapeake Bay Criteria Act in 1984. And then in 1989, the Maryland Untitled Wetlands Act. The Non-Game and Endangered Species Conservation Act, our state level ESA, did a number of things. It requires and authorizes DNR to list species that are in danger of extirpation in the state. It also requires that other state agencies use their authority to essentially work with DNR to protect, enhance, recover wildlife populations. And then finally, it authorized the DNR secretary to establish programs to conserve species. And that's really what allowed the Natural Heritage Program to become established. So I'm particularly thankful for that. It gave me a job. Our funding, we get very little state tax money or general fund money. A big chunk of our budget comes from the Chesapeake Bay and Endangered Species Fund. You may recognize it as the tax checkoff. Now that we're into tax season, please keep that in mind. We also get a lot of federal money through the State Wildlife Grants Program that the U.S. Fish and Wildlife Service administers. We also get some Pittman-Robertson Act funds, and then finally we get some endangered species funds, but all provided via U.S. Fish and Wildlife Service. In order for Maryland and every other state in the country to be eligible to receive state wildlife grant monies, we developed a state wildlife action plan. Our first one was in 2005, and then again in 2015. These are really frameworks highlighting and prioritizing what should go on in each state, what should be the focus of wildlife conservation in each state. And we are going to be working on another one coming up here soon in 2026. So, sometimes the question comes up, why do state and federal resource agencies and conservation groups focus at least to some degree on rare species? And really the first person to really articulate the link between rarity and extinction was Darwin, of all people. No, I guess not too surprisingly. And then later, well into the 20th century, in the late 1970s and 1980s, this emerging field of ecology was starting to get its toehold, called conservation biology. And Rabinowitz was one of the first to really go into more detail on that link between rarity and extinction risk. And he found that in a kind of a large analysis, he found that there were three factors that really, if you could pick any three, these were the most important that seemed to be closely related to.
My favorite moth, the Luna moth. Do you see it center up?
I'm sorry.
Luna moth.
Oh yeah, yes.
Right there at the top on the center.
Yep. And he found these three things: geographic range, abundant population size, and habitat specificity. These are really important factors to consider. So here in Maryland and other states and at the federal level, these are generally the factors that are considered when trying to assess the status. How imperiled is a species? Is state or federal listing warranted? So we look at the number of populations, how many are extant, how many are viable, their size, distribution, again, habitat specificity, the types of threats, the degree and the severity of those threats. We look at population trends, both on a historical scale, as well as on a more recent contemporary scale. And of course, the life history traits, the life history of that organism. And so by using that information, we come up with what are called state ranks. And I know you tuned in to learn about mussels, but I'm going to use birds as an example to how we assign ranks, and then I'll dovetail into mussels here in a little bit. So we have different state ranks, and S1 would indicate a highly rare species. So again, using birds as an example, a black rail is a species that's ranked S1 in Maryland. S2 would be imperiled, but not nearly as so as S1. Forster's tern would be an example. Cerulean warbler will be an example of a watch list species. And then finally down to S4 and S5. S5 is a species that is secure in Maryland. And I think we can all agree that robins are doing pretty well, and they're an example of an S5. Finally, we have other ranks that indicate a species that historically occurred here, but there's still some potential for rediscovery. Olive-sided flycatcher is arguably a good example of that. And then finally, SX is a species that is presumed gone within our state boundaries. And Eskimo curlew is clearly a good example of that. In fact, it may be extinct altogether. We could do the same thing at the global level and coming up with global ranks for every species. And so we've assigned state ranks to every vertebrate group in Maryland over the years, and we've done so for some of the better known invertebrate groups, all of our vascular plant species. So we have a pretty good catalog of how species are faring. One of the nice things about this approach to prioritizing conservation at the species level is that if there's an interest in looking at the status of a species beyond our political boundaries, perhaps range wide, all of this data that we archive, all of the occurrences of every rare, threatened, endangered plant animal is archived in a database in Annapolis, and that database in turn is shared with via NatureServe with other natural heritage programs throughout the country and in Canada. And so here's an example of black rail, and you can see all the states that are highlighted in red are states where this species is critically imperiled. And you'll see that if you look range wide, this species is doing very poorly. And this is the type of data that was used in part for the recent status assessment for this species that led to its federal listing as threatened a couple years ago. You could also look at different taxonomic groups and look at the proportion of species that are imperiled based on global ranks. And here's an interesting analysis that NatureServe did years ago. And they looked at all these different broad groups of species, from freshwater mussels and crayfishes down to ferns and butterflies and mammals and birds. And what you're looking at there, starting at if you look at the top bar where freshwater mussels are listed, it's the proportion of the freshwater mussels that occur in the United States that are globally rare or imperiled. And you'll also notice a pattern here. The top 5, they have one common theme that you may pick up on, and that is that they're all freshwater aquatic groups. So although conservation tends to focus on some of the better known, and frankly, sometimes the more charismatic, attention-grabbing species, particularly birds and mammals, some of the species groups that actually need the most attention are some of these, these understated, less studied groups of species like freshwater mussels. And this analysis showed that freshwater mussels were clearly the most imperiled faunal group in our country, with nearly 70% of all 300 or so species either extinct, endangered, or well on their way to that status. We could do something similar at the state level. And here, if you look at this graph, the bars that you're seeing are the percent of globally rare in red and the percent of species that are state rare in yellow. And if you look at the figure second from the left, you'll see that mussels rank pretty highly as a group in terms of the percentage of species that are imperiled. So this information on using state ranks and global ranks in turn are used to help determine state legal statuses. And so we have 4 state legal statuses in Maryland. First is endangered. It's the highest legal status, followed by threatened. An example of a threatened bird species would be American bittern. Black skimmers are a good example of an endangered species. We also have a status called in need of conservation. Forster's tern would be an example. And finally, this is relatively unique among states across the country in terms of what they use in their state ESAs. We have a status called endangered extirpated. Here, an example would be an Appalachian Buick's wren. And this is a species that no longer occurs in Maryland. If it is rediscovered, we can list it as endangered through emergency legislation. So rather than having a years-long drawn-out process to place it back on the endangered species list, we could do it through regulations on a much shorter time frame. We try to revise and update our official list on a pretty regular basis. We published recent revisions earlier in 2021, and these are all publicly available on our website. I hear some people tell me that these are things they keep in their glove box when they're out traveling around Maryland and birding or looking at spring ephemerals, whatever they're doing. But a lot of science, decades of work have gone into each of these publications, and we update them accordingly based on the best, most recent information in science. And just really quickly, a summary. So in terms of federally listed species, we have a total of 31. 21 are animals and 10 are plants. At the state level, we have 183 animal species that are listed, and among plants, we have 390. If you want to take a closer look at the state listed species by taxonomic group, here's a breakdown, and you can see many of them are invertebrates. Many of them are insects. We have 24 mammals, 40 birds, 11 reptiles, 8 amphibians, and so on and so forth. And among mollusks, we have 9. So after all that, finally, talk now about freshwater mussels. So freshwater mussels are in the phylum Mollusca, class Bivalvia, and it's a pretty complicated group of species. They all share the same distinctive feature of having two valves, hence bivalve, and they come together via a hinge along the dorsal margins of these shell structures. All of our native freshwater mussels belong to the order Unioida, and all of our mussels in Maryland also belong to the family Unionidae. The family Margaritiferidae includes some species found in the Pacific Northwest and further north in New England and the maritime provinces, but we don't have any representatives here in Maryland. Under the order Veneroida, it includes a pretty large group of bivalves, relatively small compared to our freshwater mussels. Fingernail clams, we have about 22 species that are native in Maryland. The other two families, the Corbiculids and the Dracenids, these are represented in Maryland and in North America by non-native species, all from Eurasia. They include the well-established, unfortunately, Asiatic clam and the zebra mussel that may be familiar to you. So just to make the obvious distinction, we're not talking about marine mussels like blue mussels or American oysters. The classification of these bivalves is based on shell morphology to a large degree. And so that includes the shape, the size of the shell, the colorations, the texture. And for many species, the internal shell morphology is very important. And it's used at higher taxonomic levels to separate, say, for our freshwater mussels, our unionids, from the dracenids or the spherids.
This is Elliptio componentis. Come to my park and I'll show it to you. Lily's going to come on March 12th.
Yes, this is the eastern
Boothby Swan Park. So anybody want to come and see this, pull it out of the water, please come.
At the global level, there are nearly 900 species, but by far the greatest diversity is in North America. And if you focus in even closer within North America, most of that diversity is in the southeastern United States. This is a little outdated, this publication, but this, if you look at each state, you can see the number of species known from that state in the upper graphic, and in the lower map, you can see the percentage of those species that are imperiled. At the time, it was thought that Maryland had 19 species. We actually have 16. There have been some taxonomic revisions. A few species were lumped. The species with the greatest number of mussels is Alabama. It says 175, but actually right now it's 180. In terms of basins, the Ohio River Basin has at least 127. The Atlantic Slope, which includes most of Maryland, there are 56 species. There's just some incredible diversity in some of our southeastern states. And largely that has to do with biogeography and the geological history of that region. It was unglaciated. It has a remarkable diversity of aquatic habitats, and a vast labyrinth of stream and river systems provide all sorts of habitats to kind of set the table for evolution. And there are also other high diversities of other freshwater organisms, everything from fishes to stoneflies to crayfish. And as you've seen a little bit here, there is some relationship between that diversity and the diversity of freshwater mussels. I will say, as a natural history buff, I can't think of any group of plants or animals that has as nearly the colorful names of freshwater mussels. And I just made a few notes of some of the mussel common names. Everything from the slipper shell to the elk toe, the sculptured pig toe, the Texas pimpleback, one of my favorites, the Georgia elephant ear, the Duck River darter snapper, the Alabama heel splitter, and finally, one, I think my favorite, the Rio Grande monkey face. So I think they've got a leg up on folks who did the nomenclature for birds and amphibians and so forth. So Taking a look at the national level, how mussels are doing, again, 72% of US mussel species are extinct, at risk of extinction or declining, and of conservation concern. Similar at the state level. Maryland has 16 native species. One or more mussels can be found in every county in the state, so they occur statewide, in a variety of stream and river habitats, and even in the upper fresh tidal portions of some of the stream systems flowing into the Chesapeake Bay. We also have some species that are well established in both ponds and impoundments, including our large municipality water sources like Triadelphia, Liberty Reservoir, Pretty Boy Reservoir, and so on. Just going to pause here and get a sip of water. So 14 of our 16 species are rare, threatened, or endangered or watch list species. 7 of those are state listed, 2 of which are also federally listed. And the federally listed species are yellow lance and dwarf wedge mussel. The stream you see on the lower right is Upper Sidling Hill Creek, one of the more important streams in Maryland for freshwater mussel conservation, and a very important area overall for biodiversity. On the upper right, you see the eastern floater, and it's one of 2 mussel species that is not rare or watch list. And then on the lower right, you'll see there's actually 3 different species there. All of those are state and or federally listed. So here's the rundown of our 16 species. Everything from the rarest, dwarf wedge mussel, yellow lance, brook floater and green floater are also very high priorities in our state. And you'll see again, 2 species in white, Elliptio campanata, or the eastern Elliptio. And then further down, eastern floater, these are ranked S5 and G5, doing reasonably well. But even in the case of eastern Elliptio, it still had a range contraction here in Maryland. You know, when you start talking about the threats to freshwater mussels, it's really a laundry list of everything that you could think of that impacts our streams and rivers, which is a fairly long list. But if you try to categorize it, you pretty much come up with, you know, half a dozen or 7 or 8 major categories. And of course, one of the most important is pollution. And it could be non-point, endpoint source pollution. Flow alteration. You might ask, what does that mean? Well, if you can think of anything that changes the natural flow of a stream or river, such as a dam or ditching and channelization, that has had a big impact on our freshwater mussels. Overexploitation. I'll talk about this in just a little more detail on the next slide. Non-native species. So these non-native bivalves like Asiatic clam and zebra mussel are highly detrimental, especially zebra mussel, to our freshwater species, freshwater mussel species. But there are other non-native species that also probably have impacts, like rusty crayfish. Climate change, of course, is having an impact in a variety of ways, some unanticipated. For our mussel species that tend to occur in fresh tidal areas, that habitat is disappearing and shrinking over time with sea level rise. We also seem to be getting an increasing severity and frequency of coastal storms, and those are impacting the watersheds that support mussels. And finally, you know, perhaps the most obvious, you know, habitat destruction and alteration, and that comes in all of its forms, and that can include, you know, urbanization. You know, there's an old saying in the world of aquatics, you know, healthy forests mean healthy streams, and having a largely forested watershed helps protect streams. But as an impervious surface expands, becomes more widespread, the hydrology can be altered, a lot of nutrient inputs, a lot of other toxins entering into the watershed.
We don't need to address gun violence, but I realized in the time set.
So just briefly about the issue of over exploitation. It's not so much of an issue here in Maryland, but it has been a major impact historically and more recently, particularly in the Ohio and the Mississippi River basins. And you can really narrow it down to three things. In the 19th century, particularly in the first half of the 19th century, there were multiple pearl rushes, and freshwater mussels, you can find pearls in mussels. It's extremely rare. I don't know what the percentage is. It's probably very, very, very small. But there were periods where people were removing mussels for no other reason than to open them up and see if they contain pearls. And that led to just, you know, quite an onslaught on mussel populations. A little bit later, in the late 1900s, mussels became commercially important. They were used in the button industry. The button industry eventually faded away as plastics became used shortly after World War II into the 1950s, but still it had quite an impact. This photo here, I'm not sure where it was taken, but that mound that you see in the background is actually an accumulation of discarded mussel shells that were used in the button industry, or were going to be used, I'm sorry. And finally, more recently, and this is an ongoing issue, is the modern pearl industry. Most of the pearls, the cultured pearls, are created through the use of freshwater mussels. And so large numbers of freshwater mussels are harvested and sent to Japan, where the flesh is removed, and then little buttons, if you will, of nacre or shell are removed, and they're used as nuclei in the culturing process. So over 90% of the pearls that you'll see in a jewelry store are you're probably seeing them because freshwater mussels somehow contributed to their existence. So some basics on freshwater mussel ecology. Kind of hinted at this earlier. Their greatest abundance and diversity is in rivers and streams. And of course, that's not surprising given that most have evolved in non-glaciated areas. They're really long-lived, and they're sedentary filter feeders. And they are remarkable ecosystem engineers, and I'll elaborate on that in just a little bit. Although I just mentioned they're sedentary, unlike oysters or say zebra mussels, which have byssal threads which allow them to attach to rocky or wooden substrates, mussels don't have any such structures, and they can actually move slowly, albeit, but they can move, and they do so by using a foot, which comes out of the interior end of the shell. And this is a mussel called the creeper, and you can see here it's moving through the substrate. You usually don't see a lot of movement in a stream or river with mussels, but you might see noticeable movement during a dry spell where mussels are moving or trying to make their way back into portions of the stream channel where there's better flows, higher dissolved oxygen, lower stream temperatures. So mussels are really important filter feeders and nutrient recyclers. They're often described as being the freshwater analog to our oysters, or even considered to be kind of like our inland oysters in terms of their ecosystem function. In a healthy stream or river, freshwater mussels can be the dominant biomass. So think about that. If you go into a healthy stream, and if you were to weigh all the mussels in that stream, they would exceed the weight of any other group of species, whether it's fish or insects. They also provide habitat for other aquatic species, and mussel beds can be aquatic benthic macroinvertebrate hotspots. So where you have a bed like you see here in this photo, the density and the diversity of caddisflies and mayflies and stoneflies can be exceptionally high here. That in turn provides food and resources for other organisms like fish. Mussels are important food sources for other wildlife species, from muskrats and otters to some bird species. And there's some fish species that prey quite a bit on them. Our freshwater drum species are good examples of that. Mussels can also be good indicators of water quality. Some of our more imperiled species are particularly sensitive to high levels of ammonia and copper. They also are unique that, they have this really long lifespan. They're the longest lived freshwater invertebrates in the world. Some can live to over 100 years. So they're kind of like these long-lived sentinel creatures. And scientists are beginning to use that sentinel aspect of their lifestyle to help look at the history of pollution in stream and river systems by analyzing toxin levels in the shell structure. So one of the great ecosystem services that mussels provide is their filtration capacity. Mussels can filter up to 10 gallons per day, an individual mussel. The photo you see here are two tanks with a similar amount of water, similar substrate. The tank on the left has no mussels. The tank on the right has mussels. And the tank both tanks were stirred up. So you had these really cloudy, turbid conditions. And after several hours, you can see the pretty immediate impact that mussels have through their filtering capacity. As a group, mussel communities can filter a very large percentage of a stream or river's volume. There's been some really interesting work done in the Delaware Basin, demonstrating that through field and lab studies. And there's also been recent estimates of the monetary value of freshwater mussels in terms of their ecosystem services. It's been estimated that they can provide about $10,000 per day per mile in filtration services compared to a wastewater treatment facility. So if you want to do the math there and consider what their annual, benefit would be, you're looking at over $3.6 million per mile. Maybe the most interesting thing beyond their ecosystem function, freshwater mussels, is their reproductive cycle. It is truly unique. They have 3 life stages: the glochidia or parasitic life stage, the juvenile life stage, and then the adult life stage. Mussel species, most are sexually distinct. You have males and females at the right time of the year males at the right time of the year will release their sperm into the water column. Mussels have 2 siphons or valves, which they take in water and then expel water. And the female, through her incurrent siphon, will suck in sperm. It will become fertilized, and then they'll be reared in a special area of the mussel called the marsupium. And then either later that year, or perhaps the following spring, depending on the species, the glochidia, or parasitic life stage, will be released back into the water column. And at this point, they need a fish host to complete their life cycle. And so these small little Pac-Man-like life stages need to clasp on to the gill filaments or the fin of a fish host. And each mussel species is specific in its fish host requirements. So for example, dwarf wedge mussel, if you're in New England, it requires in that part of its range, Atlantic salmon as a fish host. Down here, a more common species is used called the tesselated darter. So you can see how fish species play a really intricate role in the life cycle and the populations of freshwater mussels. So at any rate, once these glochidia attach to the fish, they remain there for 1 to 3 weeks. They metamorphose into a juvenile life form, which is essentially a miniature mussel. They drop off of the fish, and they then go beneath the stream bed, beneath the stream substrate, down maybe 6 to 12 inches. And there they remain for up to several years, filter feeding using their foot as a filter feeding device. And then they emerge back up to the surface of the stream bed when they're sexually mature after that 2-3 year period. So they have some extraordinary adaptations that allows them to do this. And here you can see, this is a lamp mussel, and this is a female. And she has this modified tissue to entice a fish to bite it. And that gray caterpillar-like structure in the center is called a conglutinate. And a conglutinate is a modified tissue structure that contains all of the glochidia, the parasitic life stage. And when a mussel bites down, or when a fish bites down on that, having been fooled to think that it's food, it's a prey, the conglutinate will burst, and thousands of these tiny glochidia, they're about the size of a little piece of black pepper, will then go into the water column and flush through the gill filaments and attach to the gills or the fins. And each mussel species seems to have its own form of modified tissue to maximize the probability of the glochidia successfully reaching its intended fish host. So up in the upper left is the rainbow mussel, and it has modified tissue structures that resemble a crayfish. And I wish I could get a video of this, but you could actually, when the female is trying to find a suitable host, it'll actually come out from the rocks and actually move around. It looks like a crayfish. Another example is the fan shell mussel, the next photo down, using, I guess, what is called the good old worm fishing technique. It dangles out a tissue that looks like a small worm. I think one of the most interesting approaches, though, is the catch and release method that the snuff box mussel uses. And on the left there, you can see a fish called a log perch that has its nose stuck in the snuff box. The snuff box will open its valves just ever so slightly, and what the fish will see are some mussel structures that are moving ever so gently in the stream column, and they resemble potential prey. The log perch comes over, investigates, sticks its nose into the mussel to investigate further. The female mussel then quickly clamps shut, and at the same time, she's flushing thousands of glochidia into the fish's gill filaments. And then eventually the log perch is released. And just a couple other examples, the wet fly approach with the kidney shell, and then the line and lure approach of the orange nacre mussel. You don't see the mussel in the lower photo. It would be off to the left, but you might be able to make out this almost clear, translucent thread of mussel tissue waving in the stream column. At the very end, there's what looks like a fish. And on the left are the gill filaments of a fish, and you can see those white specks each represent a glochidia that is successfully attached to that fish host. And then on the right, you can see what they look like through a highly magnified microscope. There's still a lot to learn about host fish requirements for many mussel species, including some of the mussels occurring in Maryland. This is an accumulation of the confirmed host species for the various mussels that we have here in Maryland. And they include everything from white perch to hogsucker to log perch, a few other darter species, a variety of sunfish and bass species. The Maryland Natural Heritage Program, in collaboration with other parts of DNR, especially Maryland Biological Stream Survey, have been conducting surveys for mussels throughout the state for years now. We call it the Big Quest, and it's ongoing. This information is used to help assess status, identify populations that are doing well and others that might need some help. Of course, over the years, we've hired nothing but the best here in state government in terms of folks helping us with surveys. We generally use a variety of approaches in a given stream depending on the depth of the particular stream section. Usually it's a combination of snorkeling and bucketing. In some of the deeper stream and river systems, scuba techniques are used. We also do shoreline midden searches, looking for spent shells that muskrats may have accumulated on the bank, or perhaps otters. We do time surveys, and we record all the individuals found, both live and dead, by species. For species that are higher priority, we record age and size. We do some tagging so that we can monitor growth and survival rates in the future. And we collect quite a bit of information on habitat and microhabitat. So that's a typical survey of a given stream section. And here's some of the techniques in action. The bucketing is nothing more than taking a 5-gallon bucket, carving out the bottom, and replacing it with the clear plexiglass bottom. And that it's a great way to look for mussels, to search for them in shallow streams where snorkeling techniques simply aren't practical. A little tough on the back though. We often need to use submersible flashlights because of shaded conditions, and some of our species can be quite small. This might be tough to make out, but what you're looking at there is a small eastern Elliptio, and you're able to see 2 siphons. There's an in-current siphon, and that's the siphon that's used to filter in food. Then there's the excurrent, and of course, sperm, if you're a female. And then the excurrent siphon, which is used to discard feces and other particles that don't represent food. Males, of course, use it to broadcast sperm. So taking a look at mussel species richness by watershed across the state, there are some standout areas. The areas that are brightest red are those with the greatest species richness. So you see the upper Monocacy in the middle of the state. You'll see the upper Potomac in Washington and Allegheny counties, and even some of the lower Potomac sections down in the Piedmont and even down below DC. On the eastern shore, the upper Tuckahoe River drainage is particularly important in terms of mussel diversity, as well as the Corsica River watershed. Interestingly, the greatest diversity of any 8-digit watershed in the state is found in the upper fresh tidal Potomac. And there, 14 of the 16 species known from Maryland have been documented. And so what you get there is you get this crossroads of species that are closely associated with non-tidal streams. And but when you get in these fresh tidal reaches, you also pick up some specialists that seldom occur elsewhere. They're usually found only in those upper fresh tidal areas or lower non-tidal reaches. If you were to look at population strongholds of those mussel species that are considered to be high priorities, rare, threatened, or endangered, you can see some standouts again. On the eastern shore, the Corsica, on the upper Chester, have some important mussel populations. Again, the upper fresh tidal Potomac and the upper Potomac out in Western Maryland, as well as some of its tributaries like Town Creek and Sidling Hill Creek. On the other end of the spectrum, if you were to dissect historical records, museum specimens, and try to piece together historical distributions, you'd also see some pretty obvious patterns of species loss. The Anacostia is in black, relatively high number of species that historically occurred there. But actually, I think one of our audience members could attest to this. The Anacostia may be doing better than we think and is able to support, increasingly so, some species that perhaps in decades before it wasn't able to. Another standout is that watershed you see in Washington County, and that's the Conococheague Creek watershed. The Conococheague Creek watershed may have had, beyond the upper Potomac, some of the highest diversity anywhere in Maryland, but nearly all of it is lost. And it's primarily due to just chronic agricultural impacts. And there's also a Superfund site just upstream in the Pennsylvania portion of the watershed. And so now when you go into Anacostia Creek, you seldom see live mussels. But because there's so much groundwater influence and because Anacostia Creek is overlaid by karst topography, where you have high calcium levels in the stream, the mussel shells themselves tend to persist for many, many decades after the mussels have died. So it's kind of like walking into a graveyard. You can take a look back in time. And so these are some of the stream and river systems with the greatest species loss. I mentioned Anacostia, Antietam Creek, Catoctin Creek, the upper Monocacy, even though we do have some mussel species that are persisting there, they are very low numbers. And then we have too little information to make any, really meaningful assessment of what types of losses may have occurred in other major basins, for example, in the Patapsco River or in the Patuxent River, and in the Youghiogheny River, which is in the Ohio River Basin. So our most common species is the eastern Elliptio. And you can see its distribution across the state, represented by these black dots. Those represent survey locations where this species was detected. So nearly in every county, the exception of Garrett County. And you can see areas in red, which are mostly confined to the coastal plain, are where this species seems to be doing best. And you can go into some streams where eastern Elliptio is so abundant, you could find thousands over several hundred meters of stream length. And you could find dozens of mussels per square meter. And this is perhaps one of the best streams in terms of Elliptio populations. This is the upper Tuckahoe Creek, which sits on the Caroline and Queen Anne's County border on the eastern shore. The eastern Elliptio might be one of the most challenging species to identify. If you're trying to get into mussel taxonomy and ID, you're going to have to get over this hurdle because it's the most common species in Maryland, and being able to reliably identify it is pretty important. So it comes in a lot of different shapes and color patterns, depending in large part on the habitat conditions it lives in. So if it's a relatively high gradient, cold water stream system, gets moved around a bit, you might find animals that are relatively scuffed up. Whereas if you have a stream system that's lower gradient, slower flowing, gentle currents, softer substrates, you tend to get more of a yellowish, cleaner look to the mussels. And there's eastern Elliptio on the top. They can be one of the larger sized mussels in Maryland. The mussel on the bottom is the eastern floater, and this is the other relatively secure mussel species in Maryland. And on the left is the Asiatic clam, and it's found nearly statewide. So talk about some of the higher priority species in Maryland. The dwarf wedge mussel is one of our 2 federally listed species. It's state and federally endangered. Historically, it occurred in 10 streams and rivers in our state. And at this point, it may be extant in only 4, 2 in southern Maryland, and then 2 on the eastern shore. We've been doing a lot of work recently trying to get a better handle on how well these populations are doing. And we're hoping in the coming years to do a combination of conventional survey techniques and eDNA sampling to better confirm the status of populations where we haven't seen any individuals since as recently as the early to mid 1990s. So it's been quite a while. On the lower left, you can take a look at its range-wide distribution extending from the Carolinas north into upper New England. Probably the largest population remaining is in the Connecticut River. And here is Nanjemoy Creek. This is in Charles County. And this almost certainly represents the largest, most viable population remaining in Maryland. It's a small coastal plain stream, 3rd order in size. Another high priority species is the brook floater, Elasmodon varicosa. It's state endangered. Unlike the dwarf wedge mussel, which is restricted to the coastal plain, this is found well, historically in the Piedmont, but for the most part in Western Maryland. Some of the best remaining populations are in Licking Creek and in the upper Potomac. And here's Licking Creek on the lower right. The species has gotten a lot of attention range wide lately. It had been proposed for federal listing. A listing decision by Fish and Wildlife Service was recently made though, and listing was not recommended, although there are certainly some areas that need a lot of conservation attention. You can see on the left, its historical distribution, and on the right, remaining potentially viable occurrences. So quite a range contraction, particularly the mid-Atlantic, including Maryland. But probably the main reason why it wasn't listed is because populations in the north and in the southern parts of its range seem to be doing relatively well. Green floater is a species that is currently being considered for federal listing, so it's receiving quite a bit of attention as well. This species has a similar distribution in Maryland as the brook floater. It's not found on the coastal plain, although there is a record from the Anacostia watershed. It's a vague historical record from the Smithsonian, a specimen, and we're not sure exactly where in the Anacostia. That goes back to the early 1900s. But this species is perhaps the rarest species in Maryland. It's only found in Sidling Hill Creek, a tributary to the Potomac. You can see that area in red highlighted, and along the Allegheny and Washington County border. And we also have occurrences in the upper Potomac. And here's a shot of Sidling Hill Creek. This is just great habitat for green floater. Yellow lance is state and federally threatened, rather recently so a couple years ago. This is an interesting species. It's a sand-loving species. It's associated with clean, coarse, lightish white to tan colored sand. These tend to be deep shifting sandbars. And the only population we have in Maryland is in the Hollings River in the Piedmont. Although we did find an individual in the Patuxent River, which the Hollings River flows into, right at its confluence. There's still a lot of work to be done in terms of surveys and getting a better handle on its distribution in Maryland. We're at the very northern end of its range. It's doing a little bit better in the Carolinas, but it's still warranted federal listing. The host fish species to the south are two shiners, the white and the pinewood shiner. But here in Maryland, neither one of those two species occur here. So there's still some work to be done to having a better understanding of its fish host requirements.
We do have a few questions in the chat, Jim, so let's make sure to allot time for that.
Sure. Here's the distribution of yellow lance in Maryland. The red indicating records, recent records. The black representing historical records. You'll see a red dot down near Bryan Point in the upper fresh tidal. That's an unconfirmed report. A malacologist was doing survey work in that part of the Potomac years ago, and a couple individuals were reported, but we don't have any specimens to confirm. And here's a look at Hollings River, a little muddied up. We've got a team of surveyors at work there, and you can see what it looks like on the left. We place pit tags and plastic shell tags on high priority species like this so that we can monitor them, looking at survival and growth rates and movement patterns. A few other species I'll mention here: Tidewater Mucket and Eastern Pond Mussel. These are both species that are upper fresh tidal stream and river specialists and lower non-tidal reaches. So this isn't a species you'll find in Carroll County or Allegheny County. It's they're always going to be found in and around tributaries of the Chesapeake. They have a very limited distribution as far as we know, but there's still a lot of work to do on each of these. Surveying these fresh tidal reaches is very difficult. You just can't go out there at any time. You need to pay close attention to both tides and recent rain events. And the best time to go out is during low, clear stream flows, during peak low tide. So you don't have a lot of time. It's difficult to spend a whole day searching a particular stream. Unicorn Creek on the upper left is a relatively good site for Tidewater Mucket. And then on the bottom left is a shot of the upper fresh tidal portion of the Potomac. On the other side there is Mount Vernon, to give you some perspective of where that is. Asiatic clam, again, you can see on the lower left its distribution, documented in every county in the state. Unfortunately, it's fairly well established. We have some evidence to suggest that it's associated with declines of some freshwater mussels, including dwarf wedge mussel, but there's still a lot of work to be done there. Zebra mussel has been documented in Maryland recently. It was first documented in North America in 1988 in the Great Lakes. It's since it greatly expanded its range here in North America, and it does occur in the lower portion of the Susquehanna and in the upper bay in the Susquehanna Flats. Another non-native dracenid is the Quagga mussel. It too was established in the Great Lakes in 1989. Both dracenids are native to Eurasia, and they were probably both introduced by releases of bilge water from ships arriving from ports in Eurasia here in North America. We don't have any records at this point of the Quagga mussel, but it seems inevitable that it will pop up. Just briefly, these are some of our species level and stream level priorities for doing surveys and monitoring, including most of the species on the left that I just mentioned. The upper Potomac remains an area that has received relatively little survey effort, so that needs quite a bit of work. Town Creek in Allegheny County for reasons I'll describe here in a little bit. Still quite a bit of work needed on the coastal plain and upper fresh tidal streams, and then other non-tidal portions of streams on the upper eastern shore. We've been very busy in recent years doing dwarf wedge mussel monitoring. In 2020, we completed monitoring in Nanjemoy Creek. Again, this is Maryland's best remaining population. It's a beautiful coastal plain stream, has high biotic integrity, mostly forested watershed, in absence of Asiatic clams. And you can see on the lower right, these are some of the dwarf wedge mussels we found. They've been processed and tagged, and we continue to monitor those mussels on a seasonal basis, getting some really useful information on their movements and on their survival and growth rates. Another project that we started a couple years ago is in Town Creek. Town Creek historically had a relatively rich mussel fauna. It's been diminished, we think, because of just chronic, decades-long agricultural impacts and possibly some industrial impacts in the Pennsylvania portion of the watershed. And on top of that, it flows into the upper Potomac, and that part of the Potomac has had quite a bit of impacts from a variety of sources over the last century or two. And so it's created, I guess, an impasse for any colonization, any natural colonization to occur in Town Creek. But after a lot of analysis, we believe that Town Creek can actually support an intact mussel fauna. And so we began augmenting existing populations in Town Creek in 2021 and on an experimental scale at 4 sites in different parts of Town Creek. We're going to continue those releases into 2023 at an experimental level. And if we continue to have good survival and growth rates, we're going to scale that up to potentially thousands of mussels per year throughout Town Creek and possibly into the Pennsylvania section of the stream. And we'll be closely watching to see if we can have, if we're actually getting reproducing populations of brook floater, again, a state endangered species, and Atlantic spike, another state listed species, in Town Creek. There's quite a bit of other things that we do as a program that in varying degrees has some effect on freshwater mussel conservation. We provide technical guidance to other agencies, state and federal and local, on priorities for stream and watershed restoration and protection. We do environmental review. So if we get a permit application of some kind regarding a proposed development, for example, that's something that we would review, and then we would work with local and county agencies and the applicant to avoid impacts to mussels in those stream systems. We also do a lot of review and work with our state agency partners, for example, Maryland Forest Service, Department of Agriculture, and trying to avoid impacts to activities they might be engaged in. We try to be a little more proactive and work with county and local planning agencies, working on their comp plans, trying to steer development away from watersheds that are important for aquatic biodiversity, including freshwater mussels. And we also use our data on rare, threatened, endangered species, including mussels, to help direct and prioritize land acquisition and conservation easements. So those are some of the things that we're doing as a state agency. That's about all I have, folks. Just a quick salute to all of the men and women who have helped out over the years. It's a list that's far too long to list right here, but many thanks. And then finally, thanks to my kids. They've gone out quite a few days and helped. Not when they were this young, but put a snorkel on a kid's mat on a kid's head, and they're happy as a clam, so to speak.
That is wonderful. Thank you so much, Jim. That was just amazing. I think we're all amazed, and I'm so excited about this sentinel species. And there is actually an important bill called, that mentions mussels right now, the Conservation Finance Bill, which is making it easier for private companies to help finance things. And mussel restoration projects is one of the things specifically mentioned in that bill. Want me to feed you the questions from the chat one by one?
Sure.
Okay, so the first one is, what species are native to the Anacostia watershed? Are there any in Sligo Creek or Rock Creek?
Oof. Yes to all three streams. I can't cite the species for each one, but I think the Anacostia in particular had quite a few species. It would just be some clicks away in our database for me to give you an accurate answer, but I would guess at least 6 or 7 native mussels occurred in the Anacostia historically. And I know there's a lot of work going on that watershed now, and some species have actually persisted, fared better than we thought. So cheers to the Anacostia Watershed Society.
Okay.
And cheers to your friends at Sligo Creek. I belong to the listserv. Any problem that comes up, they help. So if you send them something you want them to do, they will probably do it, seriously. You can send it to me, I'm on the list, and we'll take action.
Okay, the next question is, the map showing state by state status of a species had many blank states where there should have been one or another color to indicate how much a species was endangered. Do these states not keep track? How accurate are the data for states that do track?
Help me out there, Lily. I think I missed the first part of the question. The person was referring to the
Okay, the name is Broadwell. I can't read the rest. Can you unmute yourself, and why don't you phrase your question to them? Broadwells. Do the Broadwells? Okay, it's at 7:56 in the chat. It says, the map showing state by state status of a species had many blank states where there should have been one or another color to indicate how much a species was endangered. Do these states not keep track? How accurate are the data for states?
Okay. If a state didn't have any color at all, it means that the species does not occur there. Does that make sense? I'm trying to think of which map the person might be referring to. Why don't we come back to that, Lily, and maybe
Yeah.
person tune in.
Yeah,
And,
feel free to put more in the chat, Broadwells, if you have more. The next question is the mussels water cleaning estimate net of any pollution due to mussel excretions, or do they not excrete?
they do excrete. They excrete what are known as pseudofeces. These are little capsules of undigested detritus and undigestible materials, sand particles, things like that. So they do secrete, nutrients into the stream system, but not in a form that is nearly as harmful. In the case of ammonia, for example, they'll excrete nitrogen in a form that isn't as detrimental.
So the cleaning estimates do take into account that excretion?
Yes, yes.
Okay. How badly does it hurt mussels if you walk on them?
You can crush the shells of some species. Some species can handle that kind of stress better than others, but I think as a rule, it's not something you should try to do. So for example, when we're in the stream, we have a sense of where the mussels are. And so we try to steer our foot traffic just as we're doing what we're doing in and around where the mussels are. And we try not to make repeat visits, particularly where we have large mussel beds that could be impactful.
Okay. Is it technically illegal to possess empty mussel shells?
If it's a state or federally listed species, yes. It would kind of be like if you had the feather of an endangered bird.
Okay. Do mussels thrive in man-made lakes?
Some do. And as you might expect, these are not species that are really high on our radar screen in terms of conservation. But Loch Raven Reservoir has several species that are fairly common. I mean, the populations there are probably, well, at least in the 6-digit range, if not higher.
Okay, thank you. Is there any?
And by the way, those are artificial impoundments, and you might wonder, well, how did they get there? It's most likely the case that they got there through the stocking of game fish that had glaucidia attached to their gills or fins.
Ah,
so
cool. All right. Is there any is there any redeeming quality of zebra mussels? Do they filter and cleanse water like native mussels?
They do, and because they can be so abundant, for lack of a better term, they overdo it. So in the Great Lakes, for example, there is some evidence, quite a bit of evidence, that their filter feeding reduces plankton levels so much that it has ripple effects through the food chain.
Okay. Next question. Can we at least eat Asiatic clams?
You can try, but I wouldn't recommend it. I wouldn't recommend eating any freshwater bivalves. They can accumulate toxins, and frankly, they just don't taste very well. When I was in graduate school, a couple fellow students and I, your poor graduate students, you're tired of ordering Domino's pizza. We were at an impoundment nearby, and we collected a bucket of mussels from that impoundment. And I've collected marine mussels before, like blue mussels and clams, and have purged those and steamed them up, and they're great. So we used the same approach. So we gathered around with their cheap domestic beer and a bowl of melted butter and garlic, and we had them all steamed. We purged them overnight in the bathtub, and we sat down. We started chewing and chewing and chewing. It was like chewing on a tennis shoe, and taste wasn't that great either. That was it. So we ordered Domino's.
Well, Speaking of eating, Susan wants to know what mussels eat.
They eat plankton, detritus, diatoms, very tiny organisms and nutrients that are suspended in the column, in the stream column.
Okay. Mary mentions that mussels we have in the Anacostia include eastern Elliptio, eastern floater, eastern pond mussel, eastern lamp mussel, paper pond shell, northern lance, tidewater mucket, and alewife floater.
Awesome. I think I counted 8 species there. That's great. Thank you for sharing that.
Pat asks, when you mention ag impacts on freshwater mussel populations, would you say it's primarily sediment, fertilizers, pesticides, warming, or something else that has the biggest negative effects?
Well, it certainly includes all the above. Nutrients, if it's not the #1 issue, it's at the top. Increased sediment loads, and altered hydrology. So if you're in a watershed where the predominant land use is agriculture, and you have a relatively low proportion of forest, the stream hydrology is going to be much, much different than it would in a stream system that has intact forest. So you get streams where they're very flashy. When you have your typical thunderstorm, if you were in a forested watershed, the stream might rise, might get a little turbid, but if you're in an agricultural watershed, that stream is really going to spike. And it's going to cause really high flows. And just as a physical matter, it's difficult for mussels to maintain their position in the substrate. So they could be washed down. Now they might survive that event, but it takes energy for them to move and to reposition themselves in the substrate. And they may end up at a place that simply isn't suitable habitat, or maybe not a place where they could reproduce successfully. So hydrology is a big issue.
What did you mean by nutrients?
Well, farming often involves a lot of fertilizer use, and a lot of that washes into our streams. In agricultural landscapes, probably ammonia levels are the biggest concern.
So you mean the soil nutrients? Okay, great. Mike said, I saw there are 0 species in the upper North Branch of Potomac, as well as 0 species lost. Are you aware of any good mussel references reading of pre-industrialization upstream of the Cumberland, Maryland?
I'm not aware of any. I wish there was, but I think, the North Branch of the Potomac has a kind of a difficult history in terms of water quality. There's been a lot of industrial pollution, acid mine drainage, untreated sewage, for many, many decades. And malacologists in Maryland focus most of their work in and around Washington, D.C. These were malacologists who, worked out of Smithsonian, and they didn't get out to the North Branch too far, or maybe they knew even at that point around the turn of the century that the mussel fauna had been eliminated. But North Branch it's doing better than it was. The Luke paper mill has been shut now, and it had some significant impacts on the North Branch. Some of the industrial sources of pollution are no longer a current threat, although there's some legacy issues there with probably toxic sediments in the North Branch that have to be dealt with. So, but I think the North Branch is looking up.
Okay, that's great news. Bonnie asks, any natives found in Mattawoman Creek?
Yes. I don't have a complete list in my head, but certainly Eastern Elliptio, Eastern Floater, maybe Lampsillus radiata, the Eastern lamp mussel. Mark, you might want to weigh in. I don't know if you have any experience with doing mussel surveys in Mattawoman Creek. That's kind of
Well,
down.
the Mattawoman Creek watershed is next to the Nanjamui, so there may be a Lasmodon heterodon. We do need to look for it.
Okay, great, thank you. Do mussels filter out enteric bacteria like E. coli?
Yes, they do. Yep. They do reduce E. coli levels in streams.
Can mussels enter dammed lake from stream inflow and leave in spillway outflow and enter another stream?
They can enter a lake from an inlet stream if there's already established populations in that inlet stream. I don't think mussels can make their way beyond the dam, or at least survive. However, they were able to move beyond the dam, but could disperse if a suitable host fish is in the impoundment and it somehow makes it below the dam during dam releases.
Okie dokie. Are there any other questions that I either missed in the chat or that you have? Oh, here's something from Don Comus also. Can non-native mussels enter man-made lakes when stocked with fish from Great Lakes?
Let's see. The first way to answer it is that non-native mussels can't be transported via fish because those species don't require a fish host. So they wouldn't make their way in that way through fish stockings, but they could make their way in through, if you have, for example, a boat, like in a bass fishing tournament, and there's water in the bilge, and the bilge has juvenile life forms of zebra mussels. They could be released, and that could be a colonization event. Or maybe they're just attached to the hulls of those boats, and that could be another avenue for colonization.
I don't know much about big boats at all, but is there some way they could release, there could be laws to not let them release bilge water within so many miles of the shore or something to help with that?
I'm not familiar enough to have a good answer for that, particularly with maritime traffic. But DNR makes a great.
There are some such laws. The problem is finding the staff to enforce them.
Not how to enforce, yeah.
I do know DNR will have staff at boat launches, public boat launches, like a Deep Creek Lake.
Yeah.
Trying to keep it invasives from getting a toehold.
And by the way, Deep Creek Lake was originally a natural lake, so it still has a lot of the species it used to have before it became dammed up.
Okay, well, I'm going to open it
I
up.
do have another follow-up point to make. It turns out that if a tree falls in the forest naturally, the sedimentation that gets in the stream does not hurt the freshwater mussels because the particle size is different than the size of the food it eats. When you have artificial sedimentation, that kills the freshwater mussels because the particle size is the same size as what the mussels eat, and so they starve to death.
Did we answer that question of what?
And I can send you that study, Coker et al. I can send you that research if you want.
That reminded me of the question, did we answer what do mussels eat? Maybe I was writing something down, I didn't hear it.
I think we did.
Okay, good.
I don't think I provided a good answer for that one question, or one of the early questions about some maps that I showed. But anyways.
Does anybody want to unmute and voice a question before we close out?
Yes, Sue Muller has a quick question for you, Jim.
Okay.
I'm one of those people who has to identify everything. It's annoying. And
That's
I
good.
know it's difficult to identify mussels like from pictures and whatnot. If one gets a good picture of the top of the shell, the inside of the shell, picture with the ruler for size, are most of them not identifiable with that kind of pictures, or?
I would say the opposite. I think most of them can be identified.
Oh, okay.
Yeah. There's always going to be some questions.
Yeah. I have some pictures from along the Potomac. I've been taking pictures of shells there through the last couple years, and I just haven't done anything with them yet, but I will.
Okay. If you can go online and just Google something like a freshwater mussel shell structure, and just familiarize yourself with shell morphology. There's certain inside of a shell that are really important for identifying mussels. So if you're out there with the camera, if you could focus on those features, as well as, just the overall appearance, size, shape.
Yeah.
But I think you could probably get a name on them.
Okay. I am good with the Asiatic clam. Haha.
So
It
Lots
that
of practice there.
It's
raises an
everywhere.
important question about our workshop meeting is if anybody has an area in Maryland that they want to protect because of the freshwater mussels and because of some threat, like a development that's proposed pollution problem, and you want the Sierra Club to consider taking action, please send us. You've got this, you can send it to Jen or me or the whole group or Lily, an area that you want us, the Sierra Club, to focus on to protect.
Well, can we have a little round of applause? I asked you all to unmute so we could thank Jim.
That was fabulous.
Thank you, folks. Thank you.